Hello everyone and welcome back to MooTechie. We hope you're ready to continue our exploration of food microbiology. This session will dive deeper into the specific bacterial genera that are crucial for dairy technologists and food science students to understand. Let's begin with some important gram-negative bacteria. First we have Campylobacter. Two species, Campylobacter jejuni and Cam. coli, are foodborne pathogens. They are small, microaerophilic, helical motile cells found in the intestinal tract of humans, animals, and birds. They are mesophiles, meaning they grow at moderate temperatures. Next, we look at Pseudomonas. These are straight or curved rods. They are aerobes and motile rods, and importantly, they are psychrotrophs, meaning they can grow at low temperatures. They are found widely in the environment and include important spoilage species like Pseudomonas fluorescens, *PSC, aeruginosa, and Pse. Putida. They can metabolize a wide variety of carbohydrates, proteins, and lipids in foods. Then we have Xanthomonas. Most characteristics of this group are similar to those for Pseudomonas. They are plant pathogens and can cause spoilage of fruits and vegetables. Interestingly, Xanthomonas campestris strains are used to produce xanthan gum, which is a common food stabilizer. Another genus is Acetobacter. These are ellipsoid to rod-shaped cells. They occur singly or in short chains and can be motile or non-motile. They are aerobes and mesophiles. They oxidize ethanol to acetic acid. They cause souring of alcoholic beverages and fruit juices and are used to produce vinegar. They can also spoil some fruits. An important species is Acetobacter aceti. Moving on, we find Gluconobacter. Many characteristics of this group are similar to those of Acetobacter. Specifically, Gluconobacter oxidans causes spoilage of pineapples, apples, and pears, leading to rot. Now, let's examine Acinetobacter. These are rods that occur in pairs or small chains. They show a twitching motility because of the presence of polar fimbria. They are strictly aerobic and found in soil, water, and sewage. An important species is Acinetobacter chalcoaceticus. Next is Moraxilla. These are very short rods, frequently approaching a coccoid shape. They occur singly, in pairs or short chains and may be capsulated. They are found in the mucous membrane of animals and humans. An important species is Moraxilla lacunata. Then we have Alteromonas. Most currently assigned Alteromonas species are of marine origin and might be present in foods of marine origin. They need 100 millimolar sodium chloride for optimum growth. The species Alteromonas putrefaciens pojang, recently reclassified as Chivanella putrefaciens pojang, is important in fish and meat spoilage. They are psychrotrophs. Another genus is Flavobacterium. These are rods with parallel sides. They are non-motile, form colored colonies, and some species are psychrotrophs. They cause spoilage of milk, meat, and other protein foods. Example species is Flavobacterium aquatic. Now we come to Alcaligenes. These are rods or coccobacilli. They are motile and present in water, soil, or fecal material. They are mesophiles and cause spoilage of protein-rich foods. An example species is Alcaligenes faecalis. Next is Brucella. These are coccobacilli, mostly single and non-motile. Different species cause disease in animals, including cattle, pigs, and sheep. They are also human pathogens and have been implicated in foodborne brucellosis. For instance, Brucella abortus causes abortion in cows. Finally, in this section we have Psychrobacter. This genus was created in 1986 and contains one species, Psychrobacter immobilis. They are coccobacilli and non-motile. They can grow at 5 degrees Celsius or below, show optimum growth at 20 degrees Celsius, and are unable to grow at 35 degrees Celsius. They are found in fish, meat, and poultry products. Now let's look at gram-negative facultative anaerobes. First is Citrobacter. These are straight rods that occur single or in pairs and are usually motile. They are mesophiles found in the intestinal contents of humans, animals, and birds, and in the environment. They are included in the coliform group as an indicator of sanitation. An important species is Citrobacter freundii. Next is the very important genus Escherichia. These are straight rods that can be motile or non-motile. They are mesophiles found in the intestinal contents of humans, warm-blooded animals, and birds. While many strains are non-pathogenic, some are pathogenic and involved in foodborne diseases. Non-pathogenic strains are used as an indicator of sanitation in coliform and fecal coliform groups. The key species is Escherichia coli. Then we have Enterobacter. These are straight motile rods. They are mesophiles, found in the intestinal contents of humans, animals, birds, and in the environment. They are also included in the coliform group as an indicator of sanitation. An important species is Enterobacter aerogenes. Another genus is Edwardsiella. These are small, motile rods found in the intestines of cold-blooded animals and in fresh water. They can be pathogenic to humans, but their involvement in foodborne disease has not been shown. Next is Erwinia. These are small, motile rods that occur in pairs or short chains. They are facultative anaerobes. Many are plant pathogens and cause spoilage of plant products. Example species is Erwinia amylovora. Then we have Hafnia. These are small, motile rods. They are mesophiles found in intestinal contents and the environment. They are associated with food spoilage. Example species is Hafnia alvae. Now we look at Klebsiella. These are medium rods that occur singly or in pairs. They are motile, capsulated and mesophiles. They are found in intestinal contents, soil, water and grains. They are included in the coliform group as an indicator of sanitation. An important species is Klebsiella pneumoniae. A major pathogen is Salmonella. These are medium rods, usually motile and mesophilic. There are over 2,000 serovars, and all are regarded as human pathogens. They are found in intestinal contents and are a major cause of foodborne diseases. The species is Salmonella enterica ssp. Enterica. Next is Shigella. These are medium rods, non-motile and mesophilic. They are found in the intestine of humans and primates and are associated with foodborne diseases. Example species is Shigella dysenteriae. Then we have Morganella. These are small, motile rods. They are found in the intestinal contents of humans and animals. They can be pathogenic, but have not been implicated in foodborne disease. Example species is Morganella morganii. Another genus is Proteus. These are straight, small rods that are highly motile and form a swarm on agar media. They occur in intestinal contents and the environment. Many are involved in food spoilage. Example species is Proteus vulgaris. Now we have Serratia. These are small, motile rods. Their colonies are white or pink-red, and some can grow at refrigerated temperature. They occur in the environment and cause food spoilage. Example species is Serratia liquefaciens. Next is Yersinia. These are small rods that can be motile or non-motile and can grow at 10 degrees Celsius. They are present in the intestinal contents of animals. Yersinia enterocolitica has been involved in foodborne disease outbreaks. Another important genus is Vibrio. These are curved, motile rods. They are mesophiles found in freshwater and marine environments. Some need sodium chloride for growth. Several species are pathogens involved in foodborne disease, such as Vibrio cholerae, *vib, Parahemolyticus, and Vib, vulnificus. Others can cause food spoilage, like *vib, Alginolyticus. Then we have Aeromonas. These are small rods that occur singly or in pairs. They are motile psychrotrophs found in water environments. Aeromonas hydrophila has been suspected as a potential foodborne pathogen. Finally, we have Plesiomonas. These are small, motile rods found in fish and aquatic animals. Plesiomonas shigelloides has been suspected as a potential foodborne pathogen. Now let's discuss Rickettsias. The key genus here is Coxiella. They are gram-negative, non-motile and very small cells that grow on host cells. They are relatively resistant to high temperature, but are killed by pasteurization. Coxiella burnetii causes infection in cattle and has been implicated with Q fever in humans, especially from consuming unpasteurized milk. Moving on to gram-positive cocci. First is Micrococcus. These are spherical cells that occur in pairs, tetrads or clusters. They are aerobes, non-motile, and some produce yellow colonies. They are mesophiles, resistant to low heat, and found on mammalian skin. They can cause spoilage. Example species is Micrococcus luteus. Next is Staphylococcus. These are spherical cells that occur singly, in pairs or clusters. They are non-motile, mesophilic facultative anaerobes that can grow in 10% sodium chloride. Staphylococcus aureus strains are frequently involved in foodborne diseases. Conversely, Santa carnosus is used for processing some fermented sausages. Their main habitat is the skin of humans, animals, and birds. Then we have Streptococcus. These are spherical or ovoid cells that occur in pairs or chains. They are non-motile facultative anaerobes and mesophiles. Streptococcus pyogenes is pathogenic and has been implicated in foodborne diseases. On the beneficial side, *str. Thermophilus is used in dairy fermentation and can grow at 50 degrees Celsius. Another genus is Enterococcus. These are spheroid cells that occur in pairs or chains. They are non-motile facultative anaerobes, and some strains survive low heat like pasteurization. Their normal habitat is the intestinal contents of humans, animals, and birds, and the environment. They are used as an indicator of sanitation and are important in food spoilage. A key species is Enterococcus faecalis. Now we have Lactococcus. These are ovoid elongated cells that occur in pairs or short chains. They are non-motile facultative anaerobes and mesophiles that can grow at 10 degrees Celsius. They produce lactic acid and are used to produce many bioprocessed foods, especially fermented dairy foods. Species include Lactococcus lactis subsp. Lactis and subsp. Cremoris. Several strains produce bacteriocins with potential as food biopreservatives. Next is leuconostoc. These are spherical or lenticular cells that occur in pairs or chains. They are non-motile facultative anaerobes and heterolactic fermentators. Some are used in food fermentation, but psychrotrophic strains are associated with spoilage, like gas formation in vacuum-packaged refrigerated foods. Species include Leuconostoc mesenteroides subsp. Mesenteroides, Anishlu, Lactis, and Leu. Carnosum. Some strains also produce bacteriocins. Now let's look at Pediococcus. These are spherical cells that form tetrads. They are non-motile facultative anaerobes and homolactic fermentators. Some species are used in food fermentation, while others can cause spoilage of alcoholic beverages. Species include Pediococcus acidulactici and Pinepudi. Pentosaceus. Several strains produce bacteriocins. Finally, we have Sarcina. These are large spherical cells that occur in packets of 8 or more. They are non-motile facultative anaerobes that produce acid and gas from carbohydrates. They are present in soil, plant products, and animal feces, and can be involved in spoilage of foods of plant origin. Example species is Sarcina maxima. Now we examine some spore-forming bacteria. First is Bacillus. These are rod-shaped cells that vary widely in size and shape. They can be motile or non-motile, mesophiles or psychrotrophic, and aerobes or facultative anaerobes. All form endospores that are highly heat-resistant. Some cause foodborne disease like Bacillus cereus, or food spoilage in canned products. Enzymes from some, like *bac subtilis, are used in food bioprocessing. Another is Sporolactobacillus. These are slender, medium-sized rods that are motile and microaerophilic. They form endospores, but the spores are less heat-resistant than Bacillus spores. Their importance in food is not clearly known. An example species is Sporolactobacillus inulinus. Next is Clostridium. These are rod-shaped cells that vary widely. They are anaerobes and form heat-resistant endospores. Some are pathogens, like Clostridium botulinum and Cainsulo, perfringens, while others cause food spoilage. Some species are used as sources of enzymes. Now for gram-negative endospore-forming rods, we have D-sulfotomaculum. One species important in food is Delsulfotomaculum nigrificans. The cells are rod-shaped, motile, thermophilic, strictly anaerobes, and produce H2S. Their endospores are oval and heat-resistant. They are found in soil and cause spoilage of canned food. Coming up, we look at more gram-positive rods. First is Lactobacillus. These are rod-shaped cells that vary widely. They are facultative anaerobes and can be homo- or heterolactic fermenters. Many are used in food bioprocessing and as probiotics. Some can grow at low temperatures. Several strains produce bacteriocins. Next is Carnobacterium. They are similar in many characteristics to Lactobacilli. They are found in meat and fish, are facultative anaerobes, and can grow in refrigerated meat products. Some strains produce bacteriocins. An example species is Carnobacterium piscicola. Then we have Brocothrix. They are also similar to Lactobacilli. They are facultative anaerobes and homofermentative. They are found in meat and can grow in refrigerated vacuum-packaged meat. Example species is Brocothrix thermosfacta. Another important genus is Listeria. These are short rods that occur singly or in short chains. They are motile facultative anaerobes that can grow at 10 degrees Celsius, though the cells are killed by pasteurization. Some Listeria monocytogene strains are important foodborne pathogens. Now let's examine gram-positive, non-spore-forming irregular rods. First is Corynebacterium. These are slightly curved rods. Some cells stain unevenly. They are facultative anaerobes and mesophiles. Some species cause food spoilage, while Corynebacterium glutamicum is used to produce glutamic acid. Next is Brevibacterium. Their cells can change from rod to coccoid shape. They are aerobes and mesophiles. Two species, Brevibacterium linens and Bain-to-Bray, KCI, are implicated in the development of aroma in several surface-ripened cheeses due to production of sulfur compounds. They can cause spoilage in other protein-rich products like fish. Another genus is Propionibacterium. These are pleomorphic rods that can be coccoid, bifid, or branched. They are non-motile anaerobes. Dairy propionibacteria are used in food fermentation like Propionibacterium freudenreichii in Swiss cheese. They produce proline and propionic acid. Then we have Bifidobacterium. These are rods of various shapes, arranged in V or star-like shapes. They are non-motile anaerobes that metabolize carbohydrates to lactate and acetate. They are found in colons, and some species are used in probiotics, such as Bifidobacterium bifidum *bif, infantis, and bif, adolescens *. Based on nucleic acid homology studies, several new genera have been created. Some important in food microbiology include Tetragenococcus from Pediococcus, Vagococcus from N-group Streptococcus, Vicella and Oinococcus from Leuconostoc, Cocuria from Micrococcus, Chivanella from Pseudomonas, and Alicyclobacillus from Bacillus. Now let's group bacteria by their functional roles in foods. Bacteria are major important groups due to their rapid growth and ability to grow under diverse conditions. We group them based on similarities in characteristics. First, we have lactic acid bacteria. These produce large quantities of lactic acid from carbohydrates. Genera include Lactococcus, Leuconostoc, Pediococcus, Lactobacillus mesas, and Streptococcus thermophilus. Next are acetic acid bacteria, which produce acetic acid, like Acetobacter aceti. Then, propionic acid bacteria produce propionic acid and are used in dairy fermentation, such as Propionibacterium freudenreichii. Butyric acid bacteria produce butyric acid. Some Clostridium species like Clostridium butyricum are in this group. Proteolytic bacteria hydrolyze proteins using extracellular proteinases. Genera include Micrococcus, Staphylococcus, Bacillus, Clostridium, Pseudomonas, Ultramonas, Flavobacterium, Alcaligenes, some in Enterobacteriaceae, and Brevibacterium. Lipolytic bacteria hydrolyze triglycerides using extracellular lipases. Genera include Micrococcus, Staphylococcus, Pseudomonas, Ultramonas, and Flavobacterium. Saccharolytic bacteria hydrolyze complex carbohydrates. Genera include Bacillus, Clostridium, Aeromonas, Pseudomonas, and Enterobacter. Thermophilic bacteria grow at 50 degrees Celsius and above. Genera include Bacillus, Clostridium, Pediococcus, Streptococcus, and Lactobacillus. Psychrotrophic bacteria grow at refrigerated temperature, at or below 5 degrees Celsius. Genera include Pseudomonas, Alteromonas, Alcaligenes, Flavobacterium serratia, Bacillus, Clostridium, Lactobacillus, Leuconostoc, Carnobacterium brochythrix, Listeria yersinia, and Aeromonas. Thermoduric bacteria survive pasteurization. Genera include Micrococcus, Enterococcus, Lactobacillus, Pediococcus, Bacillus spores, and Clostridium spores. Halotolerant bacteria survive high salt concentrations up to 10%. Genera include Bacillus, Micrococcus, Staphylococcus, Pediococcus, Vibrio, and Corynebacterium. Aciduric bacteria survive at low pH, below 4.0. Genera include Lactobacillus, Pediococcus, Lactococcus, Enterococcus, and Streptococcus. Osmophilic bacteria can grow at a relatively higher osmotic environment. Genera include Staphylococcus, Leuconostoc, and Lactobacillus. Gas-producing bacteria produce gas during metabolism. Genera include Leuconostoc, Lactobacillus, Propionibacterium, Escherichia, Enterobacter, Clostridium, and Disulphotomaculum. Slime producers synthesize polysaccharides. Genera include Xanthomonas, Leuconostoc, Alcaligenes, Enterobacter, Lactococcus, and Lactobacillus. Spore formers produce spores. Genera include Bacillus, Clostridium, and Disulphotomaculum. Aerobes require oxygen. Genera include Pseudomonas, Bacillus, and Flavobacterium. Anaerobes cannot grow in oxygen. Species from Clostridium are included. Facultative anaerobes grow with or without oxygen. Genera include Lactobacillus, Pediococcus, Leuconostoc, enteric pathogens, and some Bacillus serratia and coliforms. Coliforms are used as an index of sanitation. Genera include Escherichia, Enterobacter, Citrobacter, and Klebsiella. Fecal coliforms, mainly Escherichia coli, are also used as a sanitation index. Enteric pathogens cause gastrointestinal infection. This includes pathogenic Salmonella, Shigella, Campylobacter, Yersinia, Escherichia, Vibrio, Listeria, and Hepatitis A. Because of the importance of these bacterial groups, many laboratory methods are designed to detect a specific group instead of a single species. Similarly, control methods are sometimes designed to destroy or prevent the growth of a specific group. Thank you for joining this deep dive into the world of foodborne bacteria. We've covered a vast landscape, from specific genera like Lactobacillus and Salmonella, to functional groups like psychrotrophs and spore formers. Understanding these groups, their sources, growth requirements, and roles in spoilage, disease, and production is absolutely essential for ensuring food safety and quality in the dairy and food industries. We hope this session was incredibly informative. Thank you. See you in the next class.